Good morning, class. Our today's topic is cellular organelles. Means all the organelles which are present in the cytoplasm are known as the cellular organelles. You can see in this diagram or picture that these are the organelles that found in the cytoplasm, like Golgi apparatus, smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, nucleus, mitochondria, some cytoskeleton, centriole is present in the animal cell, vacuoles present in the animal and plant both the cell, and lysosomes, the cell membrane also. So these are the organelles that found in the cell. First of all, in the cellular organelles, we have to study the outer covering of any cell, that is known as the cell wall. Actually, in the algal cell wall, it is made up of cellulose, galactans, mannans and minerals like calcium carbonate and sometimes silicates also. Other plants which have a cell wall that is made up of the cellulose, sometimes 50% cellulose is there, hemicellulose, bacteria is there, and sometimes it is made up of proteins. This is the structure of the cell wall. It is also known as the fluid mosaic model. You can see here the chain of the carbohydrate, the chain of the glycoprotein, the chain of the glycolipids, cholesterol is also there. In the cytoplasm, you can see here, this is the bilayer, fat layer, and in the center, this is the protein layer. Now, what is the function of the cell wall? Cell wall is going to be protect the cell from the mechanical support of mechanical damage, or it is support from any infection. It help to the cell formation, means interaction also with the help of the plasmodesmata. Inside the cytoplasm, we find some rod-like structures is present, that is endoplasmic reticulum, commonly called the ER. It is a very network of a tiny tubular structures that is scattered in the cytoplasm, and it is found around the nucleus. It have a double membrane boundary and occur in form of a membranous sacs-like structure. If it is formed in the sacs-like structure, it is called the cisternae. Sometimes vessels is there and sometimes tube-like structure is known as the tubules. There is two kinds of endoplasmic reticulum occurring in a cell, smooth type and rough type. You can see here, if no ribosomes is attached, it is in smooth type of surface known as the smooth endoplasmic reticulum, and if the ribosomes are attached or joined, it is known as the rough type of endoplasmic reticulum. So it have ribosomes on its outer surface, rough endoplasmic reticulum. Smooth, it have no ribosomes attached to it. Common functions of the ER is that both the type of endoplasmic reticulum serves as a channel for the transportation of the proteins, lipids and sterols inside the cell, and they are also provide some mechanical support to the cell also. The another organelle is known as the Golgi apparatus. It was first observed by the Camillo Golgi in 1898, and it is a very dense strain reticular structure that is found near the nucleus. This is the structure of the Golgi apparatus, whereas cistern-like structure, newly formed vessels is there. Lumen means the empty spaces is also there. The outgoing transport vessels which is going to transport the material. So this is known as the Golgi bodies or Golgi apparatus. The main function of this Golgi bodies are that it pack the material for the intracellular transport, as well as sometimes for the secretion to those materials outside to the cell. Many proteins synthesized by the ribosomes are modified in the cisternae of the Golgi apparatus, and it is the site of the formation of the glycoprotein, as well as sometime it is for going to form the glycolipid also. It forms the acrosome, which is of the sperm. In human sperms, there is a acrosome region which is found on the head area of the sperm cells, which have lysins or enzymes is there, which is have a very useful phenomena during the fusion of the sperm cells to the ovum cells at the time of the penetrating. Golgi cisternae form the primary lysozymes by the process of the budding. Another organelles is a spherical, sometimes irregular membrane bound vessels, sometimes bud off, forms with the bud off from the Golgi cisternae or Golgi apparatus are known as the lysosomes. Lysosomes vessels contain a large number of the hydrolysis enzymes like lipases, proteases, carbohydrases. Many enzymes is there, and all these enzymes are optimally active at the acidic pH, and hence they are known as the acid hydrolysis also. These lysosomes help in the intracellular digestion of the food material in the unicellular living organism. This is the anatomy of the lysosomes. You can see here inside there is a hydrolytic enzyme mixture is going to be filled, and the glycostated membrane is there for the transportation of the proteins. In plant cells and in animal cell, the vacuoles are present, which are the membrane bond spaces in the cytoplasm. The single membrane boundary of the vacuoles is called the tonoplast. It is found in the plant cell if there is only single boundary found in the vacuoles, and in plant cells there is a single large central vacuoles that occurs near about 90% of the volume of the cell, means the most largest organelles of the plant cell. The tonoplast of the plant cell is going to facilitate the transport of number of ions and molecules into the vacuoles against the concentration gradient, and so their concentration is significantly very high in the vacuoles than in the cytoplasm. In animal cells, vacuoles are either absent, and but in the young cells it may be very small and few in the number. On the basis of this filling material, the vacuoles types are the sap vacuoles, means vacuoles of the plant cell are called the sap vacuoles as they contain the minerals, excretory products, etc. And if the vacuoles found in the unicellular organelles like Amoeba, Paramoecium, it help in the excretion, and for the purpose of osmoregulations it is known as the contractile vacuole. Some organisms like Amoeba and Paramoeciums, the food vacuoles is also there. They are formed by the engulfing of the food material by the process of the phagocytosis, means cell eating, and pinocytosis, cell drinking is also there. So this is due to this phenomenon the food vacuoles is formed. The most important organelle is known as the mitochondria. It is a double membrane bond fused like shape structure, boat like structure, and it is a semi-autonomous organelle, means it is an autonomous organelle, means it can make all the functions necessity inside the mitochondria. Reason behind this that some DNA molecules are present in the mitochondrion. The outer membrane is smooth, but the inner membrane is having a projects in form of a mini fold like structure. It is called the cisternae, that is found into the matrix, and the matrix is rich in the enzymes because it is used in the Krebs cycle, and the circular DNA molecules and many small ribosomes like 70S subunit. 70S means 70 subunit ribosomes are present on this matrix. The main function of mitochondria is in the aerobic respiration, means when the oxygen is present for the formation of the ATP molecules. This is the picture or the 3D view structure of the mitochondria. You can see here in the cisternae, in the inner membrane, there is a DNA. You can see here there is a DNA, circular DNA is found in between the cisternaes, in between the foldings. Another very important organelles are known as the plastids. The plastids are also double membrane bond organelles of the different shapes. They are found only in the plant cell and contain the pigments and stored as the products. There are three types of plastids depending upon their color and function. Number one is the leucoplast. Leucoplast are the oval, spherical and sometimes rod-like filamentous structure. It is a colorless plastids usually found in for the purpose of the storage, like found in the tuber area and fruits in the rhizomes, etc., and mostly they are not exposed to the sunlight. The main function is to store the nutrients like starch. if it is if its starch is found in the leucoplast, it is known as a amyloplast. If protein is there, aleuroplast, and if fat is there, ileoplast is known. So depending upon the type of the reserve food material. In case of the chromoplast and chloroplast, two plasts are also there. The chromoplast are the color plastid containing the mainly the yellow color, red color and orange color pigments, means carotene and xanthophyll is there, and these are found in mostly in the petals of the flower and skin of the fruits. You can see the color petals and skin of any fruits. So chromoplast, and in the chloroplast it may be a lens shape, oval, spherical, discoid, ribbon like and cup shape organelles, and chloroplast is also a double membrane covering enclosing the colorless matrix that is called the stroma. The stroma contain a very small circular double helical DNA, ribosomes and sometimes several enzymes also. This is the 3D structure of a chloroplast. You can see here the thylakoid membrane is there in between these areas, and responsible the grenum parts and the stroma parts, two parts is there. The grenum like part is grena, and rather than the grenums all are matrix, it is known as the stroma. Another very important cellular organelles is known as the ribosomes. Actually, ribosomes was first observed by the Plastids in 1953 under an electron microscope as a very dense particle. They are particularly in form of the granular structure found attached to the surface of endoplasmic reticulum and nuclear membrane also, sometimes in a very found in a freely types or in freely inside the cytoplasm. They are made up of nucleic acid like in the RNA and protein also, and having no boundary membrane. Each ribosome consists of the two subunit, means in the ribosomal structure there is a two subunit. One is the largest unit is of the 60s and subunit of the 40s, and the whole ribosomes is of the 80 subunit. S means coefficient or Siever unit. It is the unit S coefficient or the Siever unit is. Then the ribosomes are also found to occur inside the chloroplast, and it is the unit is 70s type of the prokaryotic cells. The main function of the ribosomes is to synthesize the protein, but it means the role of the ribosomes in the protein synthesis. This is the 70s structure of the ribosomes. You can see here the two units is there, and two sides is there. One is the A site, another is the P site, and the membrane messenger RNA is found in the membrane like structure in between these two folding structure, and it is the growing peptide chain is also there, because the RNA with the amino acids is going to be provided from A site to P site. Cytoskeleton is also found in the cytoplasm, and it is formed by the elaborate network of filamentous structure, and it is made up of sometimes microtubules and microfilaments also, and because it provides the mechanical support, so it is responsible to make the definite shape and structure. It maintains the shape of the cell, regulate the distribution and orientation of the cellular organelles, and lastly it is helpful in the cell movement. Cilia and flagella are hair-like movable projections outgrowthing of a cell membrane. It may be in the movement of the cell bearing them or the movement of the fluid and structures inside the organs, while flagella help in the movement of the cell bearing them. Sometimes centrioles and centrosomes is present in the cell. The centromere is the organelles usually found near the nucleus of the animal cell, motile algal cells and sex cells of the primitive land plant. The centrioles from the basal body of the cilia flagella is there, and they give rise to the spindle formation during the cell division in the mitosis and in the meiosis. Nucleus is also present in the centrally. Robert Brown discovered and described the nucleus as a cellular organelles. Later, the Fleming gave the name chromatin to be material present in the nucleus and stained by the basic dyes also. As interface include the different following parts like nuclear membrane, chromatin, reticulum, nucleolus and nucleoplasm is there. The nuclear membranes in each eukaryotic nucleus shows a double membrane boundary, and the nuclear membrane form a barrier that is between the cytoplasm and the nucleoplasm. This is the structure of nucleus, the 3D vision of the centrally nucleus. And one nucleolus or sometimes two nucleoli is found in the nucleoplasm. And the nucleoli are the site of the active synthesis of the ribosomal RNA. Means there are three types of RNAs present. One is the ribosomal RNA that is responsible for the making of the protein synthesis inside the cellular body or inside the cell. Why this is known as the structurally and functional unit. So ribosomal unit is found there. On the basis of the centromere, means we know that each and every chromosomes have two arms, and in between the arm, both the two arms, there is a disc-like structure is called the centromere. But on the basis of the position where the centromere is found, these chromosomes are four types: telocentric, metacentric, submetacentric and acrocentric. In the telocentric, the centromere is terminal, and so the chromosomes has only one arm. It is denoting as a one arm only, telocentric. In the metacentric, the centromere is in the right in the middle, and the two arms are equal in the length. If the two arms are equal and the centrally, the centromere is present, means metacentric. In the submetacentric, a little away, this centromere is a little away from the center, so one arm is slightly longer and another is slightly shorter. In the acrocentric, the centromere is situated closer to the one end, making one arm extremely short and another arm are extremely long. You can see here in the diagram, telocentric, the centromere is there, and acrocentric, submetacentric and metacentric. So there is a four type of chromosomes. Sometimes chromosomes are known as the SAT chromosome. Actually, a few chromosomes having a non-staining secondary constrictions of a particular locations, this give the appearance of a very small fragment called the satellite, and such chromosomes are known as the satellite chromosomes or SAT chromosomes. Except all these organelles, some microbodies is also present in the cytoplasm, which are spherical and oval vessels with a single membrane boundary. For example, peroxisomes, glycoxisomes and spherosomes are very common microbodies present in the cytoplasm. So this is all about the cellular organelles. Thank you and have a nice day.